Yes, we can. Thanks. Yes.
Hello? Yeah, okay. Thanks. I apologize for not being there in person. Unfortunately, our PhD interviews spilled over and it was unavoidable. But it's great to be part of this very, very exciting initiative. And I'm going to talk today about some principles that have this big heading of an algorithmic approach to biological structure formation. And I think the two previous talks have already set it up very nicely for me to proceed over here. First of all, just to get everybody excited, here are two people who have very, very broad views of science, Freeman Dyson and Paul Nurse. These quotes, one from 20 years ago and the other one more recently, sort of indicate the fact that biology has not shied away from interdisciplinary ideas. I mean, it's had a very long history over hundreds of years. And Dyson says now, in the 20th century, in 21st century, a lot of people who are really computer scientists are doing biology. The line between mathematics and biology is not so sharp anymore. And Paul Nurse alludes to this idea that you should look at biology in terms of processes and mechanisms rather than as something static. And I think these two ideas have really been quite central to the way we've approached biological questions. Very, very broadly, for me, biology looks like this. So in the middle of the triangle, you have physics and chemistry, and those things provide the constraints. Those are the laws that are immutable, and you have to deal with how molecules work and second law of thermodynamics and so on. But the things on the outside, those are the principles: information, evolution, and I put cells over there. It's a little controversial whether all of life is made of cells, but I think these kinds of very broad principles, combined with the constraints of physics and chemistry, really allow you to think about biology very broadly. I think a lot of the questions that came up after the previous two talks are about how you jump between different scales. And the fact is that information at one scale really does propagate both up and down in biology in very, very non-trivial ways. There's no dearth of open problems. So here's a sperm whale, and we don't really know how it's put together. But we don't also completely understand how proteins are put together. And so there's really a lot of work to do. And I should say, just because we know how proteins work doesn't mean we understand biology. What's very interesting to me about this whale example, of course, is the whale is made of the same fundamental molecular building blocks as we are, but it's so different at an obvious macroscopic scale, but it's also different at all scales going down. For example, whales have a version of hemoglobin, which you and I have, which have atomic level modifications so that it's able to work with the kind of oxygen saturations that are available at depth. Okay, so changes happen at all scales in all organisms. And you really need to piece together that entire chain of causality to understand what the constraints are and how things work. So this is my big, to get people excited. So structure formation, whether you want to form a whale or form a protein, there are many, many examples in biology where structures are formed. And the idea is that you want to think about that as a process. Obviously, it's not a given structure, but it's assembled from smaller components in every case, right? And there are many ways to imagine making a structure. For example, there could be very specialized machinery that's designed to make a single specific product from a single input. Okay, it's very narrow and it's only able to do this one job. And that's not the kind of thing that biology is really built on. Biology is really built on things that are adaptable across different contexts. Simple example of that is from RNA, messenger RNA, you can make different kinds of proteins because the RNA can have different sequences. And there's a machine called the ribosome, which translates those RNA sequences into protein sequences. But that's also sort of not the most complex thing you can imagine, because it's really templated, right? It's like having a mold and making a sculpture inside that mold. If you have some version of it, you can easily make sort of a copy of it. DNA replication works in the same way. Most of biological structure formation actually happens in a more complex way. It happens because there is, I'm going to call an algorithm, and I'll describe what I mean by that in the next slide. But basically, it's a process. You take building blocks, you put them in. There's some complex time dependence and spatial dependence. All kinds of things change environmentally and at microscopic levels, and you produce some final product. We've been doing work about how that kind of algorithmic structure formation happens at the molecular level, but it's really a metaphor for how a genome creates an adult. The adult is not a copy of the genome. There's no homunculus, right? The adult is really made by a recipe that's somehow embodied in the genome in ways that we're only beginning to understand. So for me, what's an algorithm? I'll describe it by example. So in fact, in the last 10 years or so, there's been a lot of progress in using to build higher order structures. You have self-replicating clusters. You can have what I call multifarious mixtures, where you retrieve structures of various types from a single nucleus. And you can actually self-assemble, rather things that reach the meso macroscopic scale from these basic molecular interactions. How do they do it? Of course, it's a process, right? It's not templated, but it's not any old process. It's an algorithmic process. An algorithm for me is a constraint on a process where if you start from some input state, there are many ways to go because things are stochastic. And what you really do is you cut off paths that take you away from where you want to go. You encourage paths that take you where you do want to go, and then you cut off paths that take you forward from there. So you reach a target and then you don't go further. I know I'm talking very abstractly, but for example, suppose you wanted to build the Eiffel Tower just based on molecular interactions. What molecules would you make, and how would you remove these off-target paths in order to get that thing to work? And in this field, there's huge amount of open questions. There are inverse problems, what materials you program. There are simple forward problems, given these building blocks, what do they make? I'm going to give you just two very quick examples from our own work before I finish the talk. So here's some very beautiful EMs of a eukaryotic cell. Those are the kinds of cells you and I made out of. And the structure you're looking at is called the Golgi apparatus, or the Golgi body. It's really pretty complicated, and it's a snapshot of this was in my title slide. So we're interested in the Golgi, not only in what it does, but how it gets made. So what is the Golgi? If I were to abstract it out very much, this nice review covers it. The Golgi is a series of compartments within which there are enzymes that build complex molecules. And these series of compartments function like a factory production line. These molecules are handed over from one compartment to the next, to the next, to the next, in order to build a complex molecule. So the cell has sort of created the structure in order to control the assembly of molecules and cut off-target pathways. One of the things we showed a few years ago was that the creation of this cellular structure actually emerges from various prescriptions at the microscopic scale in terms of molecular interactions. I won't explain what's on the slide, but basically these graphs that I've shown here with these nodes and edges are sort of an abstraction of a cell. Each gray blob is a place in a cell, is an organelle, and the arrows show how things move around between them. And biologists have taken a lot of trouble to understand what the map of a real cell looks like. Now, it turns out that by encoding the right molecular interactions, running the system, this is a simulation, but you could also do it in vitro, you actually get structures of sufficient complexity that resemble real cells, right? So this is a sort of proof of concept that a real cell is encoded by a process at some structural level. The other work we've done more recently is shown that once you have this compartmentalized milieu, you can actually use the idea of this factory production line to create very complex molecular assemblies, these little yellow circles and blue squares and so on. These are called glycans, and they're very, very important in how cellular biology works. It's very hard to imagine how to make these complex structures to order of the right shape and size if you have just one pot, like the chemists talk about one pot. But if you have many pots and you're able to control things from one pot to the other and control the transition time, you can actually build things correctly. And again, there are inverse problems here that we can solve, and we've shown that taking an algorithmic approach is really the right way to go. So I'm going to finish with a high-level thought that none of this actually works in abstraction. I think the right kind of place to do this kind of thinking is when you're embedded in an experimental biology environment. And I think NCBS, the reason I love it is because it really allows this sort of interdisciplinary interaction between multiple groups working at multiple scales, and allows a theorist like me to jump up and down between all those scales. So I'll end over here. The little cartoon on top is a picture we commissioned a few years ago for the Simon Center where I work. On the left side is this Logo turtle, if you remember the Logo programming language. And on the right side is a real turtle, or at least a picture of a real turtle. And I think this multi-scale idea captures what we're interested in. Thank you.
Hi, Mukund.
No.
Sorry, Shravan here. Can you explain a little bit about how you think machine learning or computer
Sure.
science
Yeah.
realizing?
So as you see, I didn't put machine learning in there, and it was deliberate because we haven't actually used it. But what I think its role is not so much in the sort of application-oriented framework, but in really understanding how processes like this. So it's really solving the inverse problem. Okay. So what you have are a huge number of ways in which to design building blocks at molecular scales and design their affinities and sort of design time-dependent environmental conditions for them to grow. I mean, the space is enormous. And what you want in this case is to build structures of some given size and shape and maybe physical properties. And so far, the way that's been done is by heuristics. Okay, so people have attacked very simple structures, and they found certain principles in terms of the number, the size, the shape, some temperature, those kinds of very simple external parameters that can be used to shepherd these processes out to a final conclusion. But of course, in a cell, it really doesn't happen like that. Things are much more fast and noisy and out of control, and yet the whole structure gets built, right? So I think what we could do is, and since I'm not an expert in ML, this is all just speculation, we would like to encode these problems in ways that machines understand, show them a few examples of how it's been done, and then extrapolate to larger systems and structures and ask what kinds of building blocks would you need to achieve certain outcomes. Okay, I'm sorry I can't be more specific because I'm just getting into this, but that's the rough idea.
So, hi Mukund. Just because you have framed some of your description in the form of paths being taken and a target being achieved in some way at some point in the path, like, is there any work already using reinforcement learning in this domain, or at least problem formulation using RL?
So using reinforcement learning. Okay, I see. So training by comparing to targets and
actually some trajectories being tried out.
Right. I'm not familiar, but I'll tell you that the work in ML that's been used so far is not so much these kinds of bespoke single structures, but for example, crystal lattices with multiple components that have certain properties. People have used machine learning to identify a prescription to make lattices of given shape and size, right? And that's also, I think, it's been tested experimentally. I have to understand more clearly how reinforcement learning will work here. We don't yet have such a clear problem statement, okay, that we can jump right into a machine. And so what, in fact, Shravan and I and Srikanth and others have been trying to work on is narrow down the problem from this very abstract picture that I've put into something that is more tractable and something that is more amenable to a simple machine learning approach.
Thank you.
Okay, thanks a lot, Mukund. I think we'll go to the next speaker. Thank you for your talk.